Okay, good evening, everyone. Thank you all for joining us tonight for our latest medical science lecture titled Adapting to Life in the Liver, an Immune Cell Perspective. My name is Evelina Urso, and I'm a current medicine student here at UCL and your chair for today's event. In this lecture, Dr. Laura Pallett will be discussing how the function of immune cells in the liver is influenced by factors such as location and environment. We encourage active participation, so please feel free to use the Q&A function on Zoom to submit our questions for our speaker. For those following on Twitter, the official event hashtag is #FMS Lectures. This lecture will be recorded and a link will be sent out via email to all the attendees after the event. Before I introduce our speaker, we would like to know about our audience. So we'd like to run a poll asking you to select which group you consider you belong to. The choices are prospective students and their families, undergraduate students, PhD students, PGR students, PhD academics, patients and carers, or any others. So we'll give it a minute for everyone to be able to make their choices. Okay, so we've got our results coming in. Just over half of you are prospective students and their families. Welcome. About 9% are undergraduate students. We have some PGT students, PGR students, some PhD. 13% are academics. We have some patients and carers. Welcome. And 13% are any other. Thank you so much for everyone that voted. I am now delighted to introduce our speaker. We have Dr. Laura Pallett. Laura is an UKRI Future Leaders Research Group leader within the Institute of Immunity and Transplantation here at UCL. Laura has a PhD in viral immunology, investigating the metabolic constraints imposed on immune cells in the context of a chronic viral infection. Laura's postdoctoral work was the first to shed light on the phenotype and function of a subset of human immune cells, known as liver-resident memory T cells, TRM, demonstrating that these immune sentinels can contribute to the control of chronic viral infection. Thank you, Laura, for joining us today. Now over to you.
Thank you so much for the introduction, and actually thank you for the invite and for having me and coming along. It's great to actually be here to share some facts and figures, one of my favourite things, and some of our latest data from our lab that really underpin our current understanding of chronic liver disease. And it will become clear why I'm excited about the timing of this event in a few minutes. But because I'm an immunologist and therefore love the immune system and how it functions, this will of course be through the lens of an immune cell. So we'll be looking today from a T cell perspective, and really how these cells are adapting to live in the human liver. But I also want to launch a poll today. So in the interest of World Liver Day coming up later this week, I just want to take a moment to ask you, the audience, 2 multiple choice questions. So without cheating and Googling the answers, can you answer the first question? So globally, how many people are living with chronic liver disease? Do you think it's 5 million, less than 1 million, 1.5 billion, 150 million, or you've got absolutely no idea and you wouldn't want to hazard a guess? And then the second question I'd like to ask you is, if I told you some of the common causes of liver disease are actually in fact obesity and the associated continuing fatty diet, excess and prolonged alcohol abuse, chronic viral infections, and autoimmune conditions, what proportion of chronic liver disease do you think should actually be preventable? So either 50%, 60%, 80%, or do you think it's 90%? So I just give you a minute or 2 to respond, and then we'll see where we're at. So first, how many people do you think are living with chronic liver disease globally? And second, what proportion of the disease do you think we should be able to prevent? So, okay, so it turns out the majority of you are probably wrong for the first question. There's in fact 1.5 billion people worldwide living with chronic liver disease. And the proportion that should be preventable is in fact 90%. So we'll go into the facts and figures of that in a little bit more detail as we go through today's talk. So obviously I'm completely biased, but your liver is an incredible organ. It's a complete powerhouse. It performs over 500 vital functions, such as filtering toxins, aiding in digestion and metabolism. It's also one of your best immune system protectors. And we've already kind of alluded to this, given that the answers from the poll, but liver health often gets overlooked and people don't necessarily know the numbers. So rather impressively, your liver can regenerate itself with as little as 25% healthy tissue remaining. And in fact, in some cases, the liver can regenerate with as little as 10% remaining. And it does this within a couple of months, which means a fairly recent but hugely significant advance in the field is this concept of living donors for transplantation. So part of the liver can be removed from a living donor and transplanted into a recipient, and will in fact then regrow within a few months in both the donor and the recipient. And so therefore we have the capacity now to increase the number of available organs for those individuals sitting on the waiting list for chronic liver diseases. But as we've already suggested, each year, thousands of people are being affected by chronic liver disease. We've already seen that it's 1.5 billion living and suffering from some form of chronic liver disease, with approximately 2 million lives lost each year. So we really do need some better treatments and measures to support early detection, and prevention measures are urgently required. And actually, maybe given that in 3 days' time, World Liver Day is coming up, you could show your liver some love and consider your liver health yourself. And you could do this by checking out www.worldliverday.org, because there's plenty of information and tips to improve your liver health on that website. But we've known about this kind of crisis from liver disease for some time, especially here in the UK, as now compared to 1970, you're 4 times more likely to die from chronic liver disease than other non-communicable diseases, such as diabetes, heart attack, strokes, things like COPD, or even cancer. Or if we put that statistic in a slightly different way, deaths from chronic liver disease have now increased by over 400% since the 1970s. So I think it actually stands out as one of the glaring exceptions to the vast improvements that we as a kind of community of medical scientists have made over the past 40 years in terms of health and life expectancy for chronic disorders. It's also quite striking that liver disease is the single biggest killer in individuals aged just 35 to 49. So the most common causes of chronic liver disease, as already alluded to before we launched the poll, are things like obesity, which is now represents about 60% of chronic liver disease, alcohol abuse, so excessive and prolonged uptake of alcohol, and viral hepatitis, so things like hepatitis B and hepatitis C. So in theory, these should be preventable, but given that we have quite limited treatment options for chronic liver disease, we are reliant on changes in things like lifestyle, exercise, diet, even things like avoidance of alcohol as therapeutic strategies. So we really need to do more. And progress still needs to be made to increase both public and professional awareness of the importance of early diagnosis and continued liver monitoring. And because I love facts, here's some more for you. The liver is your second largest organ after the skin, or you could say your largest internal organ. It has an average mass of between 1.5 and 1.8 kilograms. So it really does account for a substantial proportion of your body mass. The liver has 2 major lobes, and each lobe, as you can see here from the diagram, is made up of thousands of hexagonally shaped, that's a hard word to say, these hexagon shapes made up of parenchymal cells called hepatocytes. It's an extensively vascular organ. So as you can see from this beautiful plastic cast of a human liver that depicts the tiny sinusoidal vessels, these are the small vessels that are diffusing oxygen and nutrients through their capillary walls into the underlying liver cells, the hepatocyte. So the structure and the organization of the liver has profound implications for immune cell function. The liver is quite unique in the fact that it has a dual blood supply. It receives the conventional arterial blood supply, but it also has a venous blood supply directly from the gastrointestinal tract. So unusually, the liver is receiving the majority of its blood supply as venous blood. So 70 to 80% comes directly via the portal vein into the liver. So it's therefore oxygen poor, making the liver quite a hypoxic environment for cells to live in, but it is nutrient rich as it's coming straight from the gut. About 30% of your entire blood volume is passing through the liver every minute. So as we showed on the previous slide, that extensive vascular structure is exposed to lymphocytes, red blood cells, everything that's passing through your circulation every minute. And at any one time, if we snapshot the human liver and look within it, it's packed full of immune cells. So 10% of your entire immune cell mass is in your liver at any one time, which actually equates to about 50 billion immune cells at any one time. So it's ram-jammed, absolutely packed full of lymphocytes, which are scattered through the parenchyma, or that tissue structure. And so we consider the liver a frontline immune barrier, much like the skin and the lungs, where they're constantly exposed to external pathogens or potential antigens. And so the liver is ideally poised to really deal with and detect the pathogens that are entering your body via the gut. So things that you're eating or things you're exposed to. So things like your bacteria and your microbiome, the liver is constantly dealing with those things. But as a result of that venous blood supply that's coming direct from the gut, the liver blood and the blood supply going through the liver is extremely rich in bacteria and bacterial products. And this essentially means that the liver has to decide at any one time whether to ignore or to respond. And so it represents a site of extreme tolerance, maintaining this kind of balance and equilibrium between responding to something that it's seeing, and ignoring or responding. So something could be considered a threat, or it could be considered benign. And as we saw on the previous slide with that beautiful cast, the sinusoids, so the vessels within the liver, are extremely small, they're very tight. So immune cells that are constantly flooding in through the circulation squeeze through these tiny vessels and come into very close contact with the underlying liver cells. But they also come into very close contact with each other. So the lymphocytes adapt to that environment and they're influenced by the neighboring cells. And I think this is much the same way that we think of as humans being influenced by the individuals that you come across on a daily basis. So as a lab, the Pallett lab is very interested in these big picture immunological questions of how really does the liver microenvironment impact immune cell function? And we want to know this so that we could harness the knowledge from those questions to improve therapeutic strategies for the treatment of chronic liver disease, given that the landscape isn't ideal at the moment. And to do this, we have a lab based in the Pears building. So this is the Institute of Immunity and Transplantation, which is a really cutting edge new translational immunology centre in North London, so co-situated with the Royal Free Hospital in Hampstead. And we're a T-cell centric group, so we're very interested in T cells. These are a type of white blood cell that form part of your adaptive immune system. T cells, we know, are there and engage in the killing of pathogens. So they're there to essentially control for things like intracellular bacteria, infections, and tumours. So we're really interested in understanding how they're working, how they're killing cells, how they're providing that immune memory. And we do this particularly within the human liver. So in every teaspoon of your blood, there can be up to 5 million T cells. And those T cells have one key job. And that job is to survey all of the cells of your body. So this is all 36 trillion cells of your body. Can you tell I like numbers? Which are really there. So those T cells are surveying all those cells and looking for signs of damage or for infection, or even for things like cancerous cells that are transforming and don't look normal. The immune cells, these CD8 T cells that we have in every drop of blood, actually survey the immune cells themselves, So not only are they surveying all of your lung cells, your liver cells, the structural cells of your body, they're also surveying the 1.8 trillion immune cells. And the immune cells themselves, just for those that may or may not be interested, you are carrying around 1.2 kilograms worth of immune cells, which means if you were to take all the immune system out, it probably weighs, approximately a bag of sugar or the average pineapple. So there's lots of immune cells, and we're obviously very interested in immune cells. So we're interested in CD8 T cells. This is a flavor of T cells that often get called cytotoxic T cells or serial killers. They're critical for immune defense, as I said. They're searching for viruses, they're searching for intracellular bacteria, and they're continually surveying for normal or healthy looking cells. They act as serial killers. So hopefully, let's see if we can make this video work. So they act as serial killers. So this is as you can see from the video that I'm hoping is playing, the green cell, the T cell, has been stained for using a fluorescently labeled dye and is currently surveying the surface of an in vitro tumour cell. So this is a tumour cell growing on a plastic dish. And as you can see, they can extend these bits of membrane and really cover the surface of different cells, looking to see whether they need to eliminate that cell and to remove that cell and destroy it. And the T cells do this using something called their T cell receptor. So as you can see from the diagram, that T cell receptor will recognize antigen in a specific manner on with an interaction with a protein that we call the MHC protein. And so a virally infected cell or a bacterially infected cell, for example, will display antigens. These are small proteins that have derived from the infecting agent on their MHC, which then gets presented to T cells through their TCL. And we've known for many years that fully mature T cells will travel around your body. They travel through the lymphatic system, so they can move through things like your spleen, your tonsils, and your lymph nodes, and also recirculate through the blood and through your kind of arterial circulation. So in theory, T cells are remaining on standby in your body when you need them to protect you at any one point. Over the last few decades, there's been a shift in our understanding of memory T cells. So we now know that in contrast to these circulating memory T cells that move around the body in both the blood and in the lymphatics, there's a specialized population of T cells that live within your peripheral tissues. So these were discovered in the mid-2000s, and we call them tissue resident T cells. So if we think about what the Oxford English Dictionary of resident is, it's a person, but I would argue we could replace that with thing or cell that lives in a particular place or has their home there. So resident T cells really refer to memory T cells that live long-term, are specialized to maintain immunity within particular tissues, and are completely independent of your circulating T cell pool. So these are specialized resident T cells that are retained and poised within different tissues. The liver's not unique. The skin, the lung, the kidney, all of these organs in your body have specialized resident T cells. And these T cells adapt to that local environment. And then therefore you have a population of T cells that are in the right place at the right time, with the right gear, which we'll come to in a minute, with the right attitude to really respond to the local infections they're likely to see and encounter. And we can identify these T cells because they have a different flavor on their surface. They express different molecules. So two molecules that we will be discussing are, as depicted here, the CD69 and CD183. These are just proteins that we can, that the T cell will express, and we can use fluorescently labeled antibodies to label the cells and really pull these cells apart and say these ones are resident and these ones aren't. And in my lab, we study the CD8 T cell compartment in the human liver, and particularly those that remain as long-term lodges within the liver by flow cytometry. So this is a widely used method for analyzing the expression of cell surface and intracellular molecules on cells to really determine how they look, so their phenotype, That's essentially another word for how, what they're expressing on their surface and how we can identify them and how these cells are functioning. So the technique in the instance that we use it is really based on measuring the fluorescence intensity using lasers to excite these fluorescent molecules that we stick to the cell of interest. And we can do this using a single cell suspension. So we can take human liver tissue, and for this we're indebted to our clinical colleagues at the Royal Free Hospital through the Tissue Access for Patient Benefit Initiative run by Dr. Amir Gander to obtain human liver tissue samples that are really deemed surplus to diagnostic requirement. So when as a section or explanted liver occurs, so this is when they're taken or removed as part of the necessary clinical care for individuals living with chronic liver disease, we in the lab get access to bits of that material when the donors are consenting to research. So despite where we get that tissue from, I want to just take a moment to explain these flow cytometry plots. We can stain from a different panel of antibodies to allow us to detect the flavor of the T cell. And as I mentioned previously, there's two markers, the CD69, which interferes with how a T cell would be able to egress and enter the circulation. So by expressing CD69, these cells can no longer sense or move back into the circulation. So by expressing CD69, you're likely to stay put in the tissue of origin, and CD103. So as you can see from the kind of fact plots on the far left, every dot on these graphs is an individual cell, and we look for what quadrant they fall in. And as you can see, if you look in the blood and you look in that top right quadrant, so these are cells expressing both of these retention molecules, so expressing both CD69 and CD103. So we've got two markers that cause these cells to stay put. We don't find them in the circulation, as you would expect, because they're resident within the tissue. And in the human liver, we have a population that varies in size in terms of the proportion and the percentage of these cells, but we can detect these regardless of the source. So by looking at the summary data, regardless of the source of the human liver that we sample, we can find a population of resident T cells. And we were the first to characterize and phenotype these resident T cells in the human liver. We can also use another powerful technique, which is called immunofluorescence, which allows you to visualize these cells. So from the right-hand side, we can actually use fluorescently labeled antibodies in much the same way we did before, but in tissue sections. So we can really determine the location of the T cell of interest. So by staining for the CD103 and the CD8 molecule and looking down the microscope, we can see where these cells are sitting, which cells these T cells are interacting with, who they're talking to, who they're being influenced by, and how that anatomical structure of the liver really influences where these cells sit, interact, and adapt. And this allows us to use patient material, not as a single cell suspension, but actually in a kind of localized tissue fashion. And to take up residence within a tissue, T cells have to express a number of different proteins on their plasma membrane. So that outer membrane of the cell, T cells will upregulate or start to express a number of different proteins that allow them to then interact with underlying cells, the liver stroma, for example, or the extracellular matrix. So these are the proteins that are laid down to ensure structural integrity of a tissue. So for example, we know that tissue resident T cells use some proteins that are known as chemokine receptors, CXCR3 and CXCR6. And this allows them to interact with soluble mediators or cytokines and chemokines that are produced by the cells of the liver. So by expressing the receptor, they can sense these ligands and they stay put. And in fact, we mentioned CD103 on the previous slide as being a marker that allows us to detect and identify a resident T cell. But in fact, it has a functional relationship with the underlying liver cells, because CD103 in itself binds to E-cadherin, which is the molecule expressed by all of the hepatocytes, so all of those hexagon structures that we introduced earlier. And so again, by the resident T cell expressing these molecules, it means it's stuck, it's tethered to that underlying hepatocyte and stays. And so depending on what chemokine receptors a resident T cell will express, it will really dictate the exact location that it sits within the tissue, and the profile and function that these cells take on. So why we care? We really care about these resident T cells because they're more functional. They're for want of a better word, a better cell than their non-resident counterparts. So a resident CD8 T cell produces more of these soluble mediators, cytokines and chemokines, in response to antigen. So when seeing a tumor cell, for example, presenting antigen on its MHC, these resident T cells respond much faster, making more of these antiviral and anti-tumor mediators to really allow the kind of quick elimination and killing of the infected cell. And so these are just, again, flow cytometry plots where we've isolated human liver, we've taken the cells out, we've made a single cell suspension, and we've done what we call T cell stimulation assays in a dish. So by using proteins and experimental reagents to mimic that interaction of the TCR with the MHC, we can look to see how much cytokine a T cell can make and how quickly that it can do it. And so just from the summary plot, we can see that the kind of black bar essentially is the one to look at. These resident cells make lots of cytokines very, very quickly compared to those that are circulating. So compared to those found in the blood. C cells are very good. They have the right gear. They have the right attitude because they've ensured through their chemokine receptor profile, they're going to stay put, and they've got all of the antiviral and anti-tumor cytokines ready to secrete and to help control infection. So we know from our work that these resident T cells really play a key role in controlling chronic viral infections. Our work has really shown they can control chronic hepatitis B infection. Others have shown chronic hepatitis C infection. So these cells are really very good at antiviral immunity. We know also from work of others that they can contribute to the elimination of a primary liver cancer. So hepatocellular carcinoma is the kind of key sequelae, so that kind of end point of chronic liver disease happens in a lot of individuals. And actually is one of the kind of leading causes of death in terms of cancer-related deaths rising year on year. So we know these cells are very good at antiviral and anti-tumor control. But also what's very important is the emerging role of these resident T cells in killing the fibroblasts. So in the context of fibrosis, for example, that again can be a kind of disease consequence of chronic liver disease. We know that these resident T cells could actually play a role in reducing fibrogenesis and actually reducing that progression in fibrosis. So we've got T cells and we're interested in those at the right time in the right place. But what actually happens in patients with chronic liver disease? So for years and years, we've been sampling tissue samples from a range of individuals that are living with varying chronic liver diseases. And if we just assess how many of these resident T cells there are in these individuals, just by taking the proportion of T cells that have this flavor, this resident T cell flavor, using flow cytometry as we previously described, we can plot these graphs that just have every dot of an individual patient and are showing you the proportion of TRNs or resident T cells. So from the two graphs on the left-hand side, regardless of how we categorize the severity of the disease of chronic liver disease, so we can either use steatosis, which is the presence of fat in individuals with metabolic liver disease, or we can just say healthy versus fibrosis. So those that have got the normal liver architecture, so very kind of early or no chronic liver disease, versus individuals with some level of chronic liver disease, we see that the number of resident T cells, so these presumably highly functional useful, good T cells increase in number as chronic liver disease progresses. So there's more TRMs in chronic liver disease. But interestingly, we've already said that these cells can be, in the normal context, highly functional. They can secrete more antiviral and anti-tumor cytokines, and in theory, represent a good cell. But what we know in individuals living with chronic liver disease is that when we do those short-term stimulation assays in our dish to assess the level of function, these cells are functionally impaired. So my lab, this is unpublished data from my lab, but what we're really starting to ask questions is, how do we address that? How can we reverse the functionality of these cells? How can we rescue? How can we target these cells therapeutically to give individuals with advanced chronic liver disease the right cell at the right time? How can we, get these cells back to where we want them? And we're also asking questions relating to how those cells are accumulating in these patients. Because if they're accumulating but are not functional, something's gone wrong. So these are the kind of questions we're beginning to address. But I want to talk a bit about how these cells are really influenced by the environment. Given that they live in the liver and they don't leave the liver, they stay there long term, we know they must be adapting to survive there. So much like, for example, humans adapting to the different environment you find yourself in, or as depicted here, trees, for example, growing in different landscapes, the requirements, depending on where you live, change. So a resident T cell in the skin, for example, would have a different energetic requirement, a different fuel requirement to, for example, those living in the liver or even in the lung. And for this, we have a PhD student in the lab who's really interested in asking what the metabolic fuels are. So how do these cells fuel their functionality? How are these cells turning the nutrients from the microenvironment into energy that they can use to make these cytokines, to make these antiviral mediators to really help control chronic liver disease? And without going into too much detail, just some of the things we're interested in is the concept that a resident T cell is more metabolically active than a non-resident T cell, or one that we find circulating around the blood. And we can assess this looking for the density of a nutrient transporter on the cell surface, and really look to see, well, if there's more of a nutrient transporter, they're more likely to be using that pathway, and therefore more likely to be reprogramming themselves into a survival state to learn to live in the environment. And just two examples here, we can look at uptake of the amino acids, for example, or from fatty acids. And both the resident T cells in our patients and in our healthy controls have more of these transporters on the surface. And so they've adapted some way to learn to live in the liver microenvironment. They've also adapted in a way that's quite unique. So a resident T cell becomes a master recycler. Because you're living in a tissue, because in disease, for example, the nutrient supply may be altered in some way, the resident cell, the cell that's long-lived, stuck, being retained within the tissue for a long time, has actually found a way to recycle its own proteins. And for this, the pathway is known as autophagy. So resident T cells really rely on autophagy or autophagic flux to recycle their own nutrients and to ensure their own survival. So they're not necessarily actually reliant on what's going on around them any one time. So not only do we know that they increase the density of nutrient transporters on their surface to, in theory, scavenge and bring in all of the nutrients from the tissue that they need to be self-sufficient, they're also recycling these proteins. And we know that within a tissue such as the liver, resident T cells can, because they're highly functional and they're constantly producing mediators and are constantly active, they can, in theory, acquire or harbour defective mitochondria. So the mitochondria is the kind of powerhouse of the cell, if you will. They can acquire defective mitochondria because they're so active and continually dividing and making mediators. But we know by using autophagy, these cells can recycle those defective mitochondria and actually use the proteins as well. So that the autophagy pathway, this master recycling pathway, is really critical for the long-term survival of resident T cells within the human liver. And Tregs are not just self-sufficient, they're also influenced, and I think as many of us are, by the neighbouring cell that they're encountering. So this is just a story I want to share a few slides with you before we go to some questions that we've recently published. That really talks about how resident T cells, so part of your adaptive immune system, is really influenced and perhaps reprogrammed or even re-educated by interaction with a neighbouring cell. So by bumping into and forming long-term contacts with a cell of the innate immune system, known as a monocyte or a macrophage, these cells acquire a different look, a different flavour, a different phenotype. So the study is really about membrane sharing between two cells when they come together, and these resident T cells are essentially stealing bits of membrane from the surface of the innate monocyte macrophage, recycling those proteins, and then changing their functional capacity to be a bit more innate-like and to have a kind of influence and re-education. And so before we go into the detail of the data, monocyte macrophages, as I said, are part of your innate immune system. They're there as a frontline defense for pathogens, for infection, for insult. They do this by expressing what we call pattern recognition receptors, and one of those is the LPS receptor. So, many bacteria will express LPS on their surface. The innate cell expresses receptors that can detect the presence of bacteria through their LPS receptor, and then that would trigger off a kind of immune protective phenotype. T cells are part of the adaptive immune system. They're a little bit slower to respond, and they're more antigen specific. So when these cells interact, we've now been able to show that the resident CD8 T cell has stolen bits of the membrane from the monocyte, and in fact has stolen these pattern recognition receptors, and then as a consequence of that re-education upon being so influenced by its neighbouring cell, it now begins to respond in an innate fashion. So it begins to respond more like a monocyte macrophage than a T cell, and so has a capacity to be a jack of all trades. And so to go through the data of how we found this out, we essentially when we use our flow cytometry technique, we normally stain for markers we know to be expressed. So for CD8 T cells, we can stain for an antibody for CD8, and that allows us to detect that flavor of T cell. We would stain for CD14, which is part of the LPS receptor, to detect the monocyte macrophage and the flavors of those cells. Normally, they would be discrete. T cells would have CD8, monocyte macrophages would have the CD14. When we co-stain for these antibodies by flow cytometry, we were able to detect a population of resident T cells that rather unusually and against expectation had this CD14 on their surface. We checked, they're not really there in the blood. These, as we've said, are resident cells, so they're stuck within the tissue. And in a huge range of liver samples that we have taken through our colleagues at the Royal Free Hospital, we find a varying degree of proportion of these resident CD8 T cells that have also got this CD14, they've got this LPS receptor on their surface. And we spent a little while wondering whether this was an artifact of our techniques, and whether actually we were seeing two cells that were stuck together rather than being able to discriminate the CD8 and the CD14 separately, did we in fact have two cells that were stuck together going through our machine at any one time? But we were able to image these cells and show that it's actually not two cells stuck together. It's not what we refer to as a doublet. These are single classical CD8 T cells that have this rather unusual phenotype. They have this CD14 LPS receptor on their surface. And it's not just the CD14 bit of the LPS receptor they have. We're also able to stain for other proteins that form part of the LPS receptor that we traditionally think of as a monocyte protein, TLR4 and MD2, which we don't see on the CD8 T cells that haven't got this CD14 phenotype. So we have essentially a population of CD8 T cells in the human liver that have now acquired the ability to respond to bacteria. We can look at these cells to see how they've got it, and just for interest and just to show you, this is a CD8 T cell shown highlighted. We can see CD8 in red, so to define the CD8 T cell. And we see these dots, these punctate dots of CD14, that it's stolen from that contact point when it's interacted with the myeloid cell, as it's been squeezing through that tight sinusoidal network, the vessels, as it squeezes past a monocyte in the liver, stealing bits of protein, recycling them and using them on its own membrane to change how it can respond to future pathogens. And we at UCL are able to explore these cells in vivo, so in human studies in a bit more detail. And so for this, we recruited a cohort of individuals and were able to assess whether we could see these cells in a different tissue site. So we knew from our flow cytometry that we had innate like CD8 T cells popping up in the human liver, that they could respond differently. But we didn't know if this was liver specific. We were able to look in the human skin, and we were able to ask the question of, if we change the environment, if we change the bacteria, if we change how the monocyte looks and whether the monocyte is in a hyperactivated state, so is on guard, is it more likely to bump into and interact and donate its membrane to the CD8 T cell? And for this, we raised blisters on either arm of some healthy control individuals, as you can see, that came to our clinical research facility. And then we were able to raise a blister in the presence of local E. coli to change how that microenvironment, so that we had a way of controlling how our CD8 T cell would respond. And in the presence of bacteria, so when our monocytes are hyperactivated, they're more likely to be donating their membrane, because we see more CD8 T cells stealing and acquiring, and then therefore co-staining the CD14, that LPS receptor, in the human skin, in a really cool in vivo model that gave us the full control over that local microenvironment. And just finally, what is the result of that re-education from talking to a monocyte? So T cells in theory are influenced, they're stealing bits of membrane, they're using it, but why? By stealing and by being completely influenced by the neighboring cell, they become what we call super responders. These resident T cells that have stolen this LPS receptor, not only can they now respond to bacteria, when they then see antigen through that T cell receptor, so the specific normal classical T cell receptor, they can respond by producing much more cytokine. So they become an even better resident T cell that would be perhaps more useful for surveying, those 36 trillion immune cells that are 36 trillion cells, flying around at any one time. Not only are they super responders in an antigen-specific way, they then start making a rather unusual profile of soluble mediators when they see bacteria, which T cells traditionally wouldn't respond to in this manner. So just by way of a summary to a heavy immunological talk on a Tuesday afternoon, in every tissue in your body, but particularly in your liver, there is a population that we call resident T cells that provide very specialist, local, rapid immune surveillance, so immune responses to protect your tissue. The cells that we see that become these resident memory T cells are long-lived. They adapt by recycling proteins, by taking up and scavenging as much nutrient as possible, by expressing chemokine receptors that allow them to stick long-term, to contribute and continue contributing to antiviral and anti-tumor immune responses. So controlling things like chronic viral infections, controlling the development of primary liver cancer. And so we believe, and we're really interested now in whether these resident T cells represent a kind of therapeutic target that we could somehow manipulate and intervene with for the treatment of chronic liver disease. And I obviously don't work in isolation. There's been a huge number of individuals involved in the work that we've been doing here at UCL over the number of years now. So I've been based at UCL for 10, 11 years now. I have my own team that is a growing and thriving team, but we sit in a wider team with the mainly lab. And as you can see here from our Christmas photo, we have lots of fun while we're doing our research. And with that, I'm more than happy to take any questions from the audience.
Thank you so much for the fascinating lecture. We are going to start the Q&A now. So the first question is, what is the difference between blood and non-resident T cells?
That's a really interesting point. So there's kind of still a slightly different flavor of a non-resident T cell. So we look at human blood, so we can take venous blood draws and detect CD8 T cells that continually recirculate. So these cells that are recirculate. We also compare them to a non-resident T cell within the human liver. So at any one time, T cells sluggishly move through that sinusoid and are influenced in it to some degree by the microenvironment, but not quite the same way of the circulation. So we have there's sort of an intermediate population that isn't quite resident yet, but might be. Okay.
I have a question about what makes resident T cells more functional and what causes them to release more mediators compared to their non-resident types.
Yeah, it's a really good question that we kind of investing lots of energy into at the moment. What makes them more functional, we don't necessarily know, in terms of somehow or other, once they become resident, they become poised and have the right machinery to deal with those local infections and things. We have an ongoing body of work that says actually when they steal bits of membrane, they then have a different signaling machinery and they're more likely to turn on pathways involved in survival and function. So it's a really interesting question that we're definitely interested in getting to the bottom of.
Okay, great. Next question is about whether these can be involved in pathogenesis. So the talk is about whether these T cells are beneficial, but is there any evidence that they can be contribute to pathogenesis?
Yeah, it's that classic thing of too much of a good thing sometimes. So there is definitely a growing body of work definitely in the liver, but also in the lung, that resident T cells can actually contribute to things like autoimmunity and autoimmune diseases, and actually not be, although they're in the right place for an antiviral, anti-tumor response, they're maybe not in the right place for an autoimmune response. And we also know that these resident T cells have an ability to be activated in a way that's not through the TCR, so not in a specific fashion. And if they're then responding in a non-antigen specific way, so in a way you wouldn't want them to, and not that targeted manner that you would expect, they can actually start causing damage. So somehow we need to learn more about the good side and understand the bad side to then know where to intervene and which diseases these cells would be useful for in terms of targeting.
Okay. On that note, is there any evidence that CD69 and 103 cells play a role in autoimmune hepatitis?
Yeah, so there's work from other groups that actually says yes, these cells can play a role. That still needs unpicking. Resident T cells are still quite new in terms of what we know about these cells, but in terms of individuals with autoimmune hepatitis, they do have an expansion of resident T cells. So it would be interesting to know, what they're doing in that context.
Another question is, can resident T cells stimulate B cells, and are the resident T cells stimulated by T helper cells or a separate mechanism?
Very good questions. Yes, and yes, basically. Resident T cells do everything you would expect of a normal T cell, just in a local environment, in a specific manner. So they're still helped by helper CD4 T cells. They can still interact and promote B cell and antibody responses in the way you would expect. In the liver, there's a bit of a caveat. So something I didn't share today, but the proportion of CD4 T cells in the liver is much lower than in the circulation, for example. And so CD8 T cells take on some of the kind of functional profile of a CD4 T cell to help themselves as well. But yeah, they do all those things you would expect.
Okay. We also have a general question about cirrhosis. What do you think are the most important predictors for the outcome of cirrhosis treatment?
Yeah, good question. Outcome for cirrhosis. Can you repeat the question?
What are some of the most important predictors for the outcomes of cirrhosis treatment?
Yeah, the treatment landscape for cirrhosis is a bit limited at the moment. We're reliant on, removing the underlying cause of cirrhosis. So there's lots of clinical parameters that we use, things like the level of liver inflammation, the bilirubin, etc. To understand new therapeutics, I would argue we really need to understand what the immune system is doing. So we're really trying to unpick in these patients what the immune system is doing locally. And to do that, we really have to sample the tissue. So we're reliant on our kind of clinical colleagues. But yeah, I'm not sure I answered the question, but hopefully that's interested the question that the person answering the question is asking enough and can email me if need be.
Okay, thank you. The next question is, if resident T cells become impaired as chronic liver disease progresses, could a potential treatment be to transplant healthy resident T cells into the liver of chronic liver disease patients?
Yeah, that's a really interesting point. So another line of our work is really to understand how to make these cells. So we know a bit about generating resident T cells. So we're working a little bit on the hypothesis that if we could generate them in vitro, we can give a circulating cell or a resident phenotype and then deliver this back to the patient. Would it get to the right place and stick and stay? So that's something we're doing. And then something in terms of CAR T cell therapy. So this, engineering T cells as a T cell therapy. We're working on making those resident so they stay long term and stay put and actually become the right place at the right time with the right gear. But it's a really interesting point. We do have we understand that certain cytokines and certain signals from the environment are what trigger residency. So we're mimicking that in vitro.
Are we born with resident T cells, or do some circulating T cells develop into resident T cells? Is another question.
Yeah, it's a really good question. So resident T cells by definition are memory. So at some point they have seen antigen and gone through the kind of memory response, so seen antigen proliferated and then stay. So in theory, if we were super clean, sterile, no, but in reality, yes, we're born with some resident T cells.
Okay, and as a general question, what led to your interest in this particular field?
That's a really nice, actually, it's a really lovely question. So prior to working on resident T cells, I was working on chronic viral infections and how T cells become exhausted by seeing too much antigen. So a bit like the checkpoint blockade, tumor exhaustion pathways that have really kind of revolutionized cancer treatment. I went to a conference. I think this is definitely one of those moments where you just hear something that changes your approach to something. So I went to a conference in 2014 and was really inspired by a scientist that was working on resident T cells in New York, and they presented human data of every organ apart from the liver. So I just needed to know if they existed in the human liver or not. And I've been doing it ever since.
Okay, that sounds great. I think we have time for maybe one more question. Since resident T cells appear relatively longer lasting and more powerful in their immune ability, are they part of the primary risk of organ rejection in transplants, as immunosuppressive drugs may affect them less?
Yeah, so this is an interesting thing. So we've shown in patients that have transplants that resident T cells can be long-lived from the donor and actually, may play a role in rejection. It's also some therapies that are given systemically may not actually eliminate resident cells in the way we think if they're protected, and hidden away from the vasculature. So it's definitely an area of active research.
Okay, thank you so much. We're now out of time and we'll need to conclude here. Thank you all for joining today. Before you go, we kindly request your feedback on today's session. A survey will be sent to you shortly after the event, and we greatly appreciate your input. Many thanks to everyone who shared their comments and questions, and a special thank you to Dr. Laura Pallett for delivering an excellent lecture. Wishing you all a very wonderful evening.